It's cheaper to do the way you're dealing with it. I don't know. Thank you.
So often you get it. Hello.
I'll keep my introduction very short, and I'll tell a really borrowed joke. And if anyone gets the answer, they get a prize. What's the difference between, and this is a borrowed joke, so some of you may know, a scientist and a pizza?
And what? Pizza?
It's the difference between a scientist and a pizza. A pizza can feed a family of 4. Thank you. I'm not sure Brian for it.
This evening is actually the 25th of these evening presentations, so thank you so much for being so ineffably and unlimitedly tolerant. I must also say that it's now over 40 years since I first spoke as an intermicro, a fact which I find scrotum-tighteningly embarrassing. and I really think it's incredible to think back all that length of time. It does make me realise that if I stick around long enough, and if the management here remain as open-hearted and tolerant as they've always been in the past, then I might even get to 50. And to have lectured over 50 years at a conference would be an extraordinary achievement, something I'd be very proud to do. Now, I'm going to use a remote, and if we're very lucky,
It'll work.
the battery won't go flat, and what's more, it will operate as Arthur said. Very nice to see some of you like Robert and Arthur returning after a gap of years. Also wonderful to see so many young people coming to this meeting. This meeting is not the largest, but it's the most refined and the most selective. And there is not a person. There is not a person anywhere in Intermicro who is not a shit-hot microscopist. There is no deadwood. You can ask any question of anybody and you will almost certainly get an accurate answer. And today, we are going to look at the microscope in the context of foolishness, fraud, and misrepresentation. It's interesting how often these talks manage to follow one another. This afternoon, Jan mentioned fraud in microscopy. And before that, Ted mentioned how the microscope and the microscopic image isn't part of the school curriculum. Well, my talk humbly follows what they said. So this evening, we're going to look at censoring the cell and how the media have banished the microscope. The BBC have made the world's best ever natural history programs. There is no organisation that comes near. The BBC have a natural history unit in Bristol, and they have produced the most dynamic and memorable and unique, amazing, informative documentaries. I mean, just look at the quality of the work that they regularly produce. They are utterly extraordinary. There is nobody who comes near. And of course it's David Attenborough, whom I know quite well and respect enormously. It's David Attenborough who has become the world icon who presents programs. And when we've been together, we've been snapped. On this occasion, I was presenting him with an award. We met at dinners, we meet sometimes at conferences, we meet socially. And more often than not, of course, as is always the way, you will see us simply having a laugh. And it's a curious thing that when science, and particularly microbiology and the biosciences are on television, you never see anybody laugh. They're always terribly precious and serious and down to earth and rather like preachers or philosophers. They hardly ever even break a tiny smile. And yet we know that when we meet and when we do our science, we're a laugh a minute. And the public gets an entirely spurious idea of what people actually like. So watch David's early days.
So much of the Microscope we were unable to film. Cameras, the lighting, the size of the equipment, all meant that the extremely small, the very large, the ultra-fast, or the infinitesimally slow were beyond our reach. In the last 30 years, things have changed out of all recognition. We can now film everything. The giants of the world's oceans to the smallest invertebrates.
Now that's interesting. Notice what David said. And remember, he's reading from a script. He's been encouraged by the director to say what he should say. The words are David's, but the thoughts and the editorialization is the director's. And he said how 30 years ago we couldn't do half of this stuff. How now we can film everything down to the smallest thing. And that now is possible, then wasn't possible. And yet he skates around the fact that there is this incredible gap, because the BBC has now acquired this archive of life, but there is nothing to do with microbes anywhere in their output. Nothing at all. If you log on to the BBC, they've got an entire site which is devoted to the various categories of things that they have filmed over the years. You can click onto them even to see the cats. And you see we go simpler and simpler down past the cephalopods, spiders and mites and their allies, down and down to more, as we would regard them, taxonomically lowly and simpler forms, until you get to the very bottom of the final page with sea urchins and polychaete worms. No nematodes, of course, because they'd be possibly microscopic. So when you get to polychaete worms, which are actually not simple creatures at all, and that's where it ends. If you click on algae, the only alga they've got is the giant kelp, which can be 150 feet long. If you want to see the sexual excitements of Spirogyra in the autumn, if you want to see Chlamydomonas fighting with its friends for food, then I'm afraid you will be disappointed. They simply aren't there. They just miss them out. They just do not exist. And yet you think, well, surely everybody knows what microbes are like. Why do we need television programs? They're all over the telly, and of course they are. And when you look at microorganisms on television, this is the kind of thing that you see. Microbes with silly faces on. And there's nothing very new about it, I may say. Here is an example from the Calgary Public Library site, with all the little microbes having angry-looking faces. And there's nothing new in this. Look at that circular field of view down the microscope with cartoon microbes. And now come back with me to the Victorian era. This was a drop of London water filled with microbes, and these are actually quite representative of some of the sorts that you might find, but they're all caricatures of the management who were in charge of the London water supply at that time. It's a political cartoon. And that was actually based on this very famous cartoon of life through a microscope. And again, you will recognise many of these as quite close to actual microbes, but they've all got little cartoony faces. Now, that was meant as a joke, but if you look on the Calgary site, it's got these organisms. I mean, this presumably is some kind of cholera organism, and we have ciliates and flagellates around as well, with their funny little faces and their jagged teeth. And in the bottom it says, these are what they look like magnified times 100. Nobody said they're not like this. The times 100 almost gives them authenticity. This is how the public see microbes. This is how the public see microbes. And the view of what germs are like occurs all the time on television, even in my favourite cartoon.
Are you aware that you've exposed your skin to a whole host of airborne debeautifiers? Mom, this dermal magnifier will show the extent of the damage. He's eating my beauty. Try this rejuvenating lotion. It contains over 60 ingredients. millions of them.
Now, you see, that's all good, clean fun, except that it reminds us that that caricature view of microbes is what people actually mean. It's a bizarre fact that here we are in an era, and I've said this before, I said this in a leading article in Nature in the 70s, that the public are familiar with the spiral molecule of DNA, with the craters on the moon, with the rings around Saturn. They know all this stuff. They've heard all about a living cell, and they could draw any one of them except a living cell. This is the thing which we're made of. It's the thing which predicates our life and our death and will eventually kill us, and the public don't even know what a cell looks like, although it's what they're made of. And the reason they don't is because of this tyranny of banishment of microscopic images on TV. Here's an early American advert.
Today I get to clean the bathroom bowl.
Stuck with the nastiest job in the house? Make it vanish.
Vanish?
Total Action Vanish. Try it. See, Total Action Vanish lifts off water stains, disinfects, bleaches, even leaves a clean, fresh fragrance. You just brush and flush. Next time you're stuck with the nastiest job in the house, make it vanish.
And as the years went by, we lurched unsteadily into the 80s. The same kind of thing was still found on your TV programs.
I got the bathroom bowl blues. I've been meaning to start cleaning the bathroom bowl. Oh, it's the low-downest, bluest job in town. But with tough Gringoline, you beat the bathroom bowl blues. One little pour, never need more. Water turns green, bowl still is clean. Tough Gringoline beats the bathroom bowl blues. Yeah!
Neat bit of singing, you might think.
Yeah.
Now, as the years went by, people began to realize that you could use cartoons in a kind of quasi-educational fashion, like this example.
Whoa, whoa, whoa! Aren't you forgetting something?
Look, if this is about my room, I'll clean it up after I get a snack.
I'm not talking about your room. It's your hands. You can get all kinds of gross things from dirty hands. All day long, germs get on your hands. Hand washing with soap is the single most effective way to keep from catching germs from someone else. If you wash your hands with a bar of soap, you remove 95% of the bacteria on your hands. You'll be a germinator.
Cool! I want to be a Terminator.
Not only will you be healthier, but you won't pass germs on to your family or friends. So, let's learn the right way to wash your hands.
What a charismatic little actress. I'm sure she ended up pumping gas or working McCrone.
Not to me! I'm the first germ ever to conquer this area!
Oh no! Oh, Domestos,
Extra fresh gel stripe and victory over germs flush after flush. New Domestos Rim Block. Millions of germs will die.
Now there's a Canadian one. And what about this from Czechoslovakia?
Další WC blok. Užíváte si bakterie? Jasně, šéfe. Oni si myslí, že nás to zabíjá. Co to tu voní a je to tak? Tak. Osvěžujíce.
Čože? Nový Domestos Bactistop!
Nikdy jsem než taky osvěžujíce nic!
Blázni, keď nás to zabíjá! Nový Domestos Bactistop likviduje bakterie a zároveň osvěžuje.
An update on our bathroom invasion plan, not a tool.
24 hours, our numbers have multiplied to 8 million. So, just one flush will spray our germs into the bathroom.
What? New Domestos 24-hour?
Oh, he's so thick.
Used daily for 24-hour protection from flying germs.
Now you may have noticed there was a kind of a green flash that came out of the lavatory bowl. And in the 70s, Domestos actually were showing genuine germs. They also had a TV commercial with the bigger, this time, green flash coming out of the job. And they then showed magnified droplets containing germs. Now, this is idiosyncratic in the sense that the coliform organisms you can see are imaged clearly through a scanning electron microscope and aren't moist and juicy and living as they really are. But nonetheless, they are actually germs. And there are nations where germs, actual bacteria, are put in front of the public. There was a time when we were. It's only now that it's become dumbed down and so superficial that people think that the public can't take it. Now, in other countries, where they are more philosophical, and where they put a far higher emphasis on obedience and discipline and the highest conceivable standards of education, people expect to see bacteria, actual real germs on their commercials. Look at this example from India.
होमवर्क किया?
कब का माँ?
हाथ धोया?
कब का माँ?
डेटॉल से?
दोनों हाथों में भी होते हैं कीटाणु जो कुछ ना कुछ छूने से आ जाते हैं। इनसे फैलती हैं बीमारियां। सिर्फ डेटॉल हैंड वॉश है इंडियन मेडिकल एसोसिएशन द्वारा प्रमाणित जो दे कीटाणु से 10 गुना ज्यादा सुरक्षा ताकि आप रहें हेल्दी, जी स्ट्रॉन्ग।
बीमारियों को गुड बाय किया?
कब का माँ?
चुनिए अपना डेटॉल हैंड वॉश। डेटॉल 100% sure.
And please notice that there was a microscope shown in that commercial. It was the right sort of microscope and it was correctly set up. It was being used properly, marred only by the fact that there was no slide on the stage. But otherwise it was okay. And now let's go next door to Pakistan and see what their television makes. Now, Pakistan at the moment is not exactly considered much of a friend, an intimate chum, a family member of your country or of mine. And I'm sure that as soon as the word Pakistan comes up, that you picture people swathed in grubby blankets, living in caves. Well, this is what Pakistan National Television regards as their standard television commercial about ways of controlling bacteria.
चार्ली फिर बीमार हो गया? रोज आम एंटीबैक्टीरियल साबुन से नहाए, फिर भी बीमार हो जाता है। बस अपनी ख्याली दुनिया में खोया रहता है। जरासीम ख्याली दुनिया में नहीं मरते, इसलिए अब माँ की दुनिया में, यानी नए डेटॉल सोप की दुनिया में। नया डेटॉल सोप, नए पैक्स, मैक्सिमम प्रोटेक्शन फॉर्मूला के साथ, जो जरासीम, वायरस और बैक्टीरिया से दे मैक्सिमम प्रोटेक्शन, जो कोई आम साबुन ख्यालों में भी ना दे पाए। यही नहीं, भरपूर मॉइस्चराइजर्स और मुनफरिद खुशबू, तभी तो हम रोज इस्तेमाल करते हैं। ले आई ना तुमने इसे माँ की दुनिया में? हाँ, डेटॉल सोप की दुनिया में, जिस पे है हर माँ को भरोसा। माँ की दुनिया में सब महफूज।
So if you wish to go to a country where you will see actual bacteria or advertisements about bacteria, don't go to Europe or the United States. Go to India or Pakistan or some other great nation where they value education in a way that we used to and no longer do. Now, many years ago, I used to devote much of my time to doing television programs. It's a phase you go through, like sucking your thumb or wetting your and I used to have a great pleasure in turning up on news programs. For example, in the 60s, this is a still from a 16mm film that I made of headlights in kids, and I showed it on TV. This is in black and white television days, and we discussed it. More recently, 30 years ago, Carol Vorderman, a girl on the right, came to my lab and we did a lengthy report about what the microscope can show. We shall return, we shall see a little of Carol in a moment. She's great. She spends most of her time, although she's a brilliant mathematician and a gifted popularizer of science, but she spends most of her time on television hosting game shows and house makeover shows and reunion shows, because there isn't any market for what it is that her genius mind would ordinarily have to do. We were last together 2 weeks ago in the presence of Her Majesty the Queen and Princess Anne, who came with me to see a new microscope that has been built in Cambridge. And I had been in contact with the builders whilst it was being constructed, and they said, do come and see it. And so Princess Anne had a look, and they showed her how it worked, and she turned to me and she said, well, that's a pretty impressive piece of kit. And I thought, what a wonderful Princess Anne sort of comment that really was. But Carol is great. And look at this example from 30 years ago.
L'interview de ce professeur gallois, Brian Ford, qui vous expliquera lui-même comment il a fait cette découverte. C'est une interview exclusive.
When earlier
scholars have examined the writings of Leeuwenhoek, they have been handicapped in one very important respect. They have almost always used the microfilm copies from the letters and not looked at the original letters. And I knew that Leeuwenhoek would not be the kind of man who would simply send to London letters. He wanted to show how good he was. He must have sent some specimens. The first specimens that I found were against one of the earliest letters he ever wrote to London. There were 4 packets, and 2 of them, in fact, turned out to be fine sections. These were not, as many people have said, just crude chunks cut with a knife to help Leeuwenhoek fit them onto his microscope. They were fine sections. They were exceptionally fine sections. In fact, I must admit they are sections as good as you would expect to find cut in a laboratory today.
C'est lui, le génial inventeur Anthony Leeuwenhoek, drapier et savant amateur du XVIIe siècle à Delft en Hollande. Ces petits êtres vivant dans une mare peuvent être vus facilement à l'œil nu.
En passant à côté d'un lac marécageux, j'ai prélevé un peu d'eau dans une fiole en verre. En l'examinant à travers mes lentilles le lendemain, j'ai vu de très nombreux animalcules. Leurs mouvements dans l'eau étaient si rapides et si variés que c'était merveilleux à voir.
Ainsi découvrit-il le minuscule Bursaria, à peu près de la poignée.
She names the organ.
On trouve des paramécies, un organisme unicellulaire, et c'est la première fois qu'on les voyait. Elles se déplacent en projetant des signes situés autour de leur corps. Et le halo qui entoure l'actinosphérium est en fait donné par les filaments gluants qui attrapent la nourriture. Et voici le Volvox, colonie de plantes vertes minuscules groupées en rond. Comme tout découvreur, Leeuwenhoek a souvent dû deviner ce qu'il voyait.
J'ai vu plusieurs centaines d'organismes les plus malheureux qu'on puisse imaginer. En effet, ils étaient bloqués, empêtrés dans leur queue, et ils se débattaient en s'étirant violemment pour la dégager.
Forme dans du vinaigre avarié, pensant que c'était des minuscules anguilles, il faisait une découverte formidable. À Londres, ces observations surprenantes sont testées et confirmées par le maître anglais du microscope.
Confirmed by Robert Hooke. And here is his original microscope we got out for them in London.
Les lentilles. L'œilleton, une lentille à l'intérieur du cylindre, et en bas, l'objectif dans un tube de cuivre. Une lentille condensatrice et un bocal rempli d'eau captent la lumière d'une lampe à huile.
Notice that the lamp is burning to provide light.
Ce qu'il a vu au travers du microscope l'a ravi. Ainsi, il a inventé la beauté de la puce.
Le microscope montre clairement que la puce est entièrement revêtue d'une armure noire, curieusement polie et hérissée de fines pointes. La tête est embellie par des yeux noirs, vifs et ronds, et 2 antennes tactiles et olfactives.
Now, you will, I hope you'll forgive me letting that run in French, but it's just a beautiful piece of film. We managed to put together a series of 16mm movies of Cartesium and Volvox and the other organisms that gave the viewer an impression of what it was that these people were really looking at. And when the Robert Hooke microscope appeared, there it is with the lamp lit. It's been shown exactly as it would have been used. That's the same as the microscope that Robert Hooke portrays in Micrographia, published in 1665. It was a beautiful film, but that's 30 years ago, when I was active in this field, and people were very happy for there to be more microscopic stuff on TV. But let's come forward away from Leeuwenhoek to Robert Brown. This is the piece I published in Nature when we showed how Robert Brown's observations were done, and those of you who didn't fall asleep at the time will remember my description of all of this at Intermicro in the 1980s, again 30 years ago. Now this research is all over the place. Everybody now knows how Brown did his work. It's in books, encyclopedias, and if you Google it, up comes this Ford research with boring and unimaginable monotonies everywhere you look. Until, of course, until the BBC decided they would lurch unsteadily into this territory. And they produced a programme, you notice it's titled Knowledge. The BBC now have a department of knowledge, They have a head of knowledge, George Entwistle, who earns $350,000 a year for doing his job. And this is the sharp intake of breath from the envious Americans in my book. And this was a series about the atom, and they decided that they would include Robert Brown's research in it. And it was odd, because I knew they were making this program, and we never had a phone call or an email. And this, ladies and gentlemen, do make sure that your seat backs and tray tables are returned to the upright and locked position, and that your seat belts are firmly fastened, because this really will produce an effect on what you think of science on television.
In 1827, a Scottish botanist called Robert Brown sprinkled pollen grains in some water.
Not on the slide, and under a microscope.
What he found was really strange, because instead of the pollen grains floating gently in the water, they danced around furiously, almost as though they were alive. For nearly 80 years, Brown's discovery remained a little-known scientific anomaly. Then, Einstein changed everything. In one staggering insight, Einstein saw that Brownian motion was all about atoms. The pollen will only jiggle if they were being jostled by something else. So Einstein said that the water must be made of tiny atom-like particles, which themselves are jiggling and are continually buffeting the pollen. If there were no atoms, then the pollen would stay still.
Now, dear Jim Al-Khalili, from beginning to end, has given a complete load of completely misleading account. Firstly, the pollen grains don't move, it's the particles inside the pollen grains. Secondly, he didn't sprinkle it on a fish tank. He put it on a slide under his microscope. Thirdly, the microscope is not being used in the way that Brown would have been using it, and indeed it is a modern microscope when Brown looked through the microscope. And the piece of video that you saw earlier is clearly of particles of Indian ink photographed under oil immersion or something of the sort, and has no relationship to Brown, has no relationship to pollen, has no relationship to Brown's microscope or anything he did in the 1820s. And finally, we have expensively commissioned graphic art which actually shows pollen grains moving about in a way that they never, ever did. Now then, people kept saying to me, and there are at least 20 of you in this room, why is it that you don't just go back to the beam and just do some programs on cells? And this was the proposal that I put to them. It was going to be a sequence of the cell. This is only, of course, a working title. And I laid it on thick. These visually beautiful television documentaries will give you, for the first time, a natural history of the living cell. Well, as I mentioned in a previous talk, this went through a number of iterations and discussions with the BBC. And eventually they came back and said that firstly they wouldn't be doing a series on the cell, and if they did it would be a one-off program and on a minority channel, and anyway they didn't think they'd do it. The whole tenor of their response was rather negative and downbeat. So I was a bit surprised when a producer friend rang me a few months later and said, oh, the BBC are doing a series of programs on the cell. This of course is an entire coincidence. It has nothing whatever to do with the fact that I put this proposal in. And in order to make sure nobody did plagiarise it, I sent it to Mark Thompson, a long-standing colleague who is the Director General of the BBC. I thought if it goes to Mark, nobody can pinch it. So I was a bit surprised, having put that in, to see that a year or so later, this was the series they decided they'd produce. Now, of course, they have a 6-figure budget. I have asked Mr. Entwistle what the cost of the series was, and he says, well, it's not BBC policy to release that kind of information. They do release the fact that he earns $250,000, however. And this is what they produced. They came back to Robert Brown, and they filmed a sequence in which they were going to do the research, which I've lectured on at this meeting and at many other countries around the world, of looking through the microscope and seeing what Brown saw. And this is what the BBC had the cheek to make of our revered hero, Robert Brown.
Decided to peer into the heart of the plant cell, and there he'd reveal something as important as the discovery of the cell itself. Now, the orchid was a lucky choice as it happened, because it has cells which are larger than other plants. If it weren't for this, it's unlikely that Brown would have discovered what he did. Now, I'm going to try and see what was so important using Brown's own microscope. Brown noticed a distinctive shape within each cell. It was a turning point in science. He called it the nucleus.
Now again, we have a complete fiction. The cells, and I saw some skeptical responses here when it was said, the cells of an orchid are not smaller than the cells of other plants, are not much larger than the cells of other plants. The seeds of orchids are the smallest seeds of any of the angiosperms. They are much smaller, for example, than Lycopodium spores are. And the cells of many orchids are extremely tiny. However, be that as it may, they are now going to recreate the experiment that I carried out in the 1980s and widely published, and you shall now see what the BBC's 6-figure budget got them. I was astonished. I thought, well, it's so easy to do. At least I didn't find it very difficult. But this is what they managed to achieve. Here comes their technical wizardry, their top cameraman, their leading microscopical expert, and this, ladies and gentlemen, is what goes on the television programme as an example of what Robert Brown could see through his microscope.
You know, Brian, I got a better image of that from last year.
Do you know, I believe you did.
Yes, I did.
Now, I mean, here is a still frame. It's better on the small screen. But here is a still frame of what they saw. Now, the image to which the esteemed Mr. Haddix is alluding is this one, which is the same specimen that I imaged through the same lens on the same microscope, and which I was honoured to win a prize for last year. If you put the two together, here you can see my own amateurish, ill-informed and quickly run-off experiment alongside the BBC's magnificent technical achievement. And the point is, of course, that people are left with the belief. I mean, the viewing dozens, for it did go out on a minority channel, were left with the belief that these early microscopes couldn't see stuff. When it was reviewed by Adrian Gill, A.A. Gill, in the Sunday Times in London, the premier reviewer of television programmes, he said, rather naughtily, "So we're off with this young, cheeky, chappy science bloke in a red shirt and a cheap suit with his enthusiasm phaser on stun." And then he says, "It couldn't escape from the central problem that the story of the cell is also the story of microscopes," and wait for it, "and that looking down microscopes is not great telly." He goes on, "The story of cells actually is interesting, it's simply not very exciting. And no amount of wishful thinking is going to make it so." Now, of course, A.A. Gill doesn't know whether it makes exciting television because he's never seen it made into exciting television. And nor have the television producers. Television producers don't know what they want. They only want what they already know.
And actor Dr. Brian J. Ford from having a long-lasting impact on science.
Well, he's done research on mad cow disease and even had laws changed because of his discoveries. Now, we had a chance to speak to him when he was in Singapore to deliver a distinguished visitors lecture at the Singapore Science Centre.
Well, one of the key studies that you have done, and that's made really headlines in the world of science, is intelligent cell. What did you mean by that?
Cells do extraordinary things. If you look at cells under the microscope, which of course people don't do these days people are looking at the minutiae, they're looking at the molecular genetics of cells, they're looking at cell biology, they're looking in the tiniest way at the smallest bits. When we were in school, we all learned of amoebae as being the simplest, most lowly form of life. But there are some amoebae, and I'm doing studies of these at the moment in England, there are some amoebae that construct a home for themselves. They pick up things like tiny specks of sand and cement them into a beautiful vase-shaped house in which they live. And sometimes you can look at cells under the microscope and see that they've been damaged. And the way that they repair themselves by actually carrying out reconstruction work is much more than just a simple chemical system. Some cells within the cell have a tiny eye.
You know, when science is explained so easy in terms that we can easily understand, isn't it fascinating?
Yeah. Well, it was very kind of it to say so. That was about an 8-minute item, and I just picked out a couple of the better goals. to use the current soccer metaphor. But the point is that, yes, it is exciting stuff. It just never gets on TV. Now, it completely amazes me that if, for example, they're going to make a period drama and a ringed dove, a collared dove, comes and sings in the background, they had to take the shot again because that species wasn't in Britain at the time that Bronte was writing her novels. If they're doing a reconstruction of some drama and a steam train comes along, they have to make sure they've got the sound effect of the right steam locomotive, or people will write it in their hundreds and complain. But with science, with the microscope, you can say any complete drivel that you wish, because the public are so disenfranchised from it, as Ted said this afternoon, Theodore said this afternoon, that there is nobody watching who's going to correct them. This isn't dumbing down. It is kind of implied censorship. Now, Leeuwenhoek is a good case in point, because the chap who presented the programme about Brown also made a sequence about Leeuwenhoek. And I invite you to watch this. Also, please note that when Hans Lonker, who is well known to many of us as a manufacturer of replicas, I never met Hans, though we've often emailed in the past, and do notice that when the presenter says, "So what am I looking at here?" that he says, "At the wrong side," which I must admit is one of the great moments in this programme.
Van Leeuwenhoek built a staggering 247 microscopes, a new one every couple of months for over 50 years, and told nobody how he made them. We've placed a drop of lake water onto a slide that slots into the device. With a bit of luck, I'll be able to see what Van Leeuwenhoek saw. What do I actually do? Where am I looking?
At the wrong side.
Yeah, I can see green.
There is a focusing knob.
There's a focus.
Yes, there is a focusing knob. This might be made...
So I can move it actually away from my eye.
This one, this is the focus.
Let me try that off. Yeah, that works. That really works. It's now in focus. Oh, wow. Oh my God, you can actually see moving creatures.
Yes.
Oh God, that's incredible. There's a tiny, tiny bug in there, which is scooting around, which I guess is a protozoa. That's astonishing.
Tiny, tiny bug in there scooting around. He's very good on the descriptive side. Now, what is interesting about that sequence is that those chlorophyte filaments that you could see, and the little protozoan or rotover, whichever it was, they were not actually as clearly seen in the shot as they are to the naked eye. If you get a little bit of pond water in a file and look at it, just look at it, damn it, and you will see the organisms clearer. The resolution, though not the magnification, the resolution is greater. And knowing how much money the BBC had spent on this ridiculous series, I thought, how cheaply could you do it? So I once picked up at a Target store a 99-cent webcam. It's only VGA resolution. It has a very low refresh rate. And so I took the same kind of algae and the same kind of microorganisms, and this was the result that we got. So this experiment cost 99 cents, ladies and gentlemen. And of course you can see the algal filaments quite clearly. And you can make out the organisms inside very well. I mean, this kind of lamentable ignorance is so utterly extraordinary. And it terrifies me to think that the public are being fed a continuing drip, drip diet of this negative and ill-informed and ludicrous view. In fact, we're being so caught up now, as I said in the Singapore interview that you saw, we're now so caught up with molecular biology and genetics. Genetics, of course, has done so much to tell us about who we are and where we came from and what we might die of. It has done nothing whatever to cure anybody, very much of anything. I mean, the great opening of vistas of new therapies that everybody was assured would follow, which is why we were pumping billions into the field, has simply failed to eventuate. But people never think of living things anymore because they don't see science like this. And this wasn't taken through a Hans Lonker replica microscope, but through a ×20 lens from a Victorian botanical microscope. A really cheap lens, I mean, a really cheap lens and a 99-cent camera. And the results there are 1,000 times better than the results the BBC managed to obtain. If you want to allow me to show off for just one little moment, using a single lens microscope properly, this is Stylonychia. This is a single lensed microscope. This is one lens the size of the head of a pin, ladies and gentlemen. The amount of chromatic aberration is minimal, and I invite you to just look at that beautiful view. That is what Leeuwenhoek could see. Now, this is an extraordinary revelation. This is a quite majestic sight, is it not? And yet this is what the television won't show you because they are so detached from this area of knowledge. I mean, the point of having a department of knowledge is presumably, firstly, to have knowledge, secondly, to have people who've got knowledge, and thirdly, to tell people who haven't got knowledge to go and get some. If it doesn't do those things, I'm at a loss to know what it's there for. So now let's come to the pièce de résistance, Leeuwenhoek's observation of spermatozoa. Now, this is what I took through a single lens microscope, and I showed it at this meeting 30 years ago, 25 years ago at least. And it's a single spermatozoan, and that is taken with a single lens microscope. Here is squamous vaginal epithelial cells, and you can even see the middle piece and the head and the tail of the sperm. It's a sort of a dried smear. Leeuwenhoek didn't look at dried smears, but I just wanted to prove that the thing could actually be resolved. This is what they look like under phase. This is positive phase contrast image of spermatozoa. And that's the kind of thing that a modern microscope will show you. So how then did the BBC come along and do their experiment? Now, Leeuwenhoek used post-coital secretions in order to find spermatozoa. He thought that masturbation would be not the right thing to do, and so he mentioned that he would not abuse himself, but that he discovered sperm in the fluids that were generated by the natural processes of loving one's wife. Quite right too, because the spermatozoa are much rarer if you look in post-coital fluids. What our cheeky chappie in the cheap suit and the red shirt did was to tell people, he said, I wasn't so modest when I got my sample of semen, and he waved it at the lens, whilst 40 or even 50 viewers would have been completely put off their toast.
Well, you know, there they are, tiny little atoms swimming along, kind of like tadpoles.
There they are, he said. Where exactly?
His own seaman, Van Leeuwenhoek, would have seen the same thing, thousands of tiny little animalcules powering along with whiplash-like tails.
Now you can see the occasional hint of movement in that somewhere, but I love the way that he says Leeuwenhoek could have seen the same thing powering themselves along with their whiplash tails. I see no superabundant evidence of whiplash tails. So here's a piece of film that I took with a single lens microscope, and this will show, look, you'll see it better on the small screen, but this will show you what a single lens microscope can actually reveal. Now this is the astonishing thing. What I've repeatedly realized in my research is that the single lensed microscope, the microscope of the late 1600s that Leeuwenhoek made, could have been used to make most of the great discoveries that were later made with optical microscopes. And the fact that you can see spermatozoa as clearly and as accurately and as lively and dynamically as that. I mean, if I put that picture on the screen up on a meeting and told you what it was, I don't think anybody in the audience would raise their hand and say, I'll bet that was taken with a microscope in the 1600s. Indeed, I have seen some much worse photographs taken with more modern instruments.
What are these of?
Sorry?
The image you just showed, what are these of? These
They're-
are having of human sperm.
Yes, they are.
They are?
Yes.
Well, they have no resemblance to what human sperm look like.
But they jolly well better have. They've sired 6 children so far. So there was a report. There was a report in Laboratory News. This is a controlled circulation magazine. It goes to every science lab and every scientist in Britain. And so they put their appalling picture with my version and what the modern microscope would show, and wrote a very pleasantly light-hearted piece to suggest that the Beebs, the BBC, were actually, let us say, neglecting their duty somewhat and failing to show people what you really can see. Then they did a series called The Story of Science by a man called Michael Mosley, who began apparently as an economist and trained as a doctor, then became a science producer and is now a science presenter. We'll see you in a moment. It became a BBC series and also a book. And in it, I looked up cell, and I turned to the page, and there was the double-page spread to show people what a living cell is like inside. And here it is, metal-plated, inside the cell. I mean, nothing like this, of course, exists in nature. It is simply a graphical model of what the double helix of DNA is like, and also sprayed with gold lacquer. There is in this book no study at all of living cells. There are some plant tissues used as wallpaper on the background of one of the spreads, but otherwise there isn't anything like a living cell anywhere in this book. And the program is just as bad.
The microscope. One of the first to use it had been Robert Hooke in the 17th century. His book, Micrographia, contains illustrations of a hidden world. The microscope had revealed the intricate structure of plants, snowflakes, and natural fibers.
Now, I do want you to notice the way in which he handled the book. These books, and I have copies of the original Micrographia in England, these books are consulted by lying them on a table, on a cloth, on a rug, perhaps on a cushion, or on a sponge foam support, like a wide open obtuse V. And the pages are turned with enormous care. Some people use cotton gloves. I don't myself, and I'm not sure it's necessary with ancient papers. But nonetheless, that is the kind of way that it's done. The way that he handled the pages, just look at the way he's handling it in one hand, then he rips open this priceless thing, and the way he turns the pages sounds like a US telephone directory being consulted. You really can't begin to conceive of the damage that is done to a book by handling it in such a profoundly inappropriate manner.
The cell finally resurfaced in the mid-19th century in the research laboratories of Prussia. Even with the best microscopes, this is all they can see. A nucleus in a translucent mush.
Now, I'm very offended at the translucent mush. The argument is, of course, he's moving towards the fact that Prussian scientists were going to discover stains. It's not quite true. It was William Perkin, the English scientist, who discovered aniline dyes. But it is true that a lot of the research work on early staining was done in Germany. But it is not the case that you had to stain something in order to study it. With the rare and infrequent example of in vitro staining, stains were only used to show particles within dead cells. And as I've often said, studying histological preparations and stained dead microbes is like trying to deduce human sociology through the study of charred and painted corpses. It simply isn't the way that you should look at cells. And the reason that you don't see the nucleus in this cell properly is because, firstly, it's not in focus. Secondly, the condenser is wrongly positioned. Thirdly, the iris diaphragm is too wide open. And the substage is being overflooded with light. It needs to be shut down properly. And curiously, I did exactly this demonstration 30 years ago with Carol, whom I mentioned to you before, when she came to my lab to do a film.
The onion will give us a living cell. Just take a piece of it and use that scalpel to pull away a bit of the top surface of the onion.
Right.
That's right.
Yeah. And then what do I do?
Just strip it back towards you. Now then, you see that little tiny thin bit that's come away, lay it down there.
That's the bit that's quite slimy when you cut off onions.
How dare you speak of plant tissues like that? Yeah, I get a drop of water. Now I'm using a pipette, but any old common old garden eyedropper would do for that.
Right.
And then with a little cover slip on top, like so. If we pop that under here, in place of the potato, we will see life as it is lived in the raw. Inside each cell, there's one on the left and two on the right there, you can see the nucleus, which of course is the controlling part of the cell. Let's just see if we...
Right in the middle.
That's right. Just see if we can find a better one. Now that is a particularly good cell. It's a perfect, typical example of a living cell.
Now, the point of that was not to show the latest microscopy or whatever. It was to try and show people that using a terribly simple microscope with nothing other than water and an eye dropper to mount specimens in, that anybody could see stuff. And programs like this had quite a good reaction. If we put the two pictures together, there on the left is the one that I got with my hastily set up microscope and deliberately eschewing any form of staining or phase or anything. It's just a light field shot. And on the right is the picture that we saw that the BBC produced with their massive budget. Now, it is appalling to my mind that people can actually take and distort real biology in this way and then propagandize this negative and destructive view to the public. The obsession with molecular biology has made everybody think that living cells are little nanobots, that they're like little electronic digital circuits. The notion of them being sentient beings who take decisions and lead their lives is hardly ever seen in the media. Yeah, that was the BBC. Now, Channel 4, which is the independent channel, but the sort of upmarket independent channel, they decided they would do a series on the genius of Britain. And they featured a man I've lectured with in the past, Richard Dawkins, a man who, of course, insists that God is just a mirage and Darwin is really the bee's knees. And until you step back and you realise that he is so angry about God and so fervent in his support of Darwin. Darwin was a great naturalist and a wonderful author, but he was not the first person to think of survival of the fittest. And survival of the fittest certainly is not the only mainspring of evolutionary progress. And in fact, I believe that Dawkins's fervour proves the opposite of what he says. I believe it proves that there is in many people an overwhelming desire to believe in something, and his God is Darwin. And the fervency of his belief in Darwin is exactly equivalent to the way in which the religious right would believe in God. I think he's just replaced one God with another and hasn't shown that the God delusion exists at all. However, we're going to see Dawkins talking about a Flea and Robert Hooke's microscope. Notice that when the microscope appears, we'll see this in the next sequence, that they don't light the flame, they use a torch. What a cop-out that is. And I want you to see how Dawkins, the evolutionary biologist, handles the specimen. He's given a jar containing human fleas preserved in 70% alcohol. And so what would you do? Well, you would pick a loop, or possibly you would platinum loop, or possibly you would take a pipette or forceps and lift it up. If you were in the countryside, you might snap off the stem of a piece of grass and lift it up. Not Dawkins, he shoves his hand inside the jar.
On the tip of my finger is a tiny flea. You almost can't see it with the naked eye at all. And that's the way it was in the 17th century. All people knew about fleas was that they caused itchy red spots and sleepless nights, until Hooke put one under his microscope.
Now, it's quite true that when Hooke put stuff under his microscope, he showed people wonderful new sights. Yes. But it's not true that people could hardly see the flea. These are specimens of parasites mounted in the 1700s. There is a flea in an ivory slider between two circles of mica held in place with brass countersteps. Here's a headlouse. You can't see much, but of course you can see them, and people knew quite a lot about fleas. In fact, I'm one day going to do a presentation into Micro on the microscopical studies that were done of tiny insects and worms before microscopes were invented, just to remind us of the extent to which people with keen eyesight and diligence were able to see things even without microscopes. That's going to be an interesting talk. I only hope I survive long enough to write it. So here is Dawkins.
This is an extremely precious object. It's one of the very early compound microscopes,
dating from the 1670s. It was designed, but not made, by Robert Hooke himself. There would have been a wick there, lit with oil coming from this reservoir here, and this great big bowl of water acted as a large lens, focusing the light through this smaller lens here on the stage where we put a flea. And instead of the oil lamp, we've got a modern torch here, which I'm just going to switch on. What he saw must have taken his breath away. A revelation from an unknown world. I can imagine it must have been a wonderful experience for Hooke and people like him to look down a compound microscope for the first time, and it opened up a whole new world of the very small. Whenever he found something new, Hooke would put it under his microscope, and slowly and painstakingly he began to draw what he saw. For the first time, we could begin to understand the innermost workings of nature. And in 1665, he published his masterpiece, Micrographia, with its astounding images and Hooke's speculations about the laws that might govern the universe. It's the most magnificent book. It was an instant sensation. Samuel Pepys stayed up all night reading it, not surprisingly, because it's full of incredible images that nobody had ever dreamt of. This flea, which is an exquisite drawing by Hooke, shows amazing detail. You see all the hairs on the legs, little hooks at the ends of the feet, which are used to cling on to the host.
I may say Samuel Pepys often stayed up all night, and in his diary, which he wrote in code, he often said whom he stayed up all night. But on this occasion, he actually did sit up all night long reading Hooke's book. He was absolutely astonished by it. And notice that Dawkins at least has the courtesy and the knowledge and the insight to handle the copy of the book correctly. This is what he saw. And of course he was so wrong when he said people gasped when they saw all this detail by looking through the compound microscope, because this is what the compound microscope shows. And yet the drawings that Hooke published in his book show details you cannot see with the compound microscope. And of course, as I've explained at meetings in the past, this is what a flea looks like. It's from the Lush Lawn site. It's the nicest macrograph I've ever seen of a flea. This is actually a cat flea, not a human flea, but the differences are slight. And if you put Hooke's drawing on the right with the modern view through his compound microscope on the left, you can see that it, of course, was a simple microscope that was used. And this is that flea specimen on the slider that I mentioned to you, which I have here imaged through a single lens microscope. And of course, you can't see any of these hairs and chitinous projections when you look through a compound instrument, but through the simple lens, the single lens microscope, which Robert Hooke described the construction of in his book, you can see it. So there are all sorts of subtle inaccuracies and mistakes in the program as we saw it. Here is a lunch at Cambridge where I'm sitting, and over here you may recognise behind his computer screen Stephen Hawking, the brief history of time man, and I was dining there because it was the time when I was nominally saying I was going to retire, and Stephen typed out to me on his Americanized auto box, I won't retire, I think I can avoid it, he said. And indeed, they did have Stephen out to actually help to narrate the final episode in Channel 4's television series.
We began this story in the 17th century, at a time of witchcraft and superstition. We've seen how a handful of men began by asking questions. What keeps the stars in the sky? What makes an apple fall? What invisible worlds exist on our skin and under our noses?
In the 350 years since, British scientists have learned to picture the enormity of the universe. And they've split the atom.
They've discovered how to cross oceans and fly at supersonic speeds. How to power turbines and light up cities.
They've uncovered the greatest secrets of life and shown us just how astonishing the world is.
Scientists have helped us live longer, healthier, and more fulfilled lives than ever before.
Above all, these men have changed our world forever. Indeed, they've made it what it is.
They were often awkward and contentious characters, people who kept on asking questions and didn't settle for second-rate answers.
Their stories explain why science is so important to us.
What is clear is that we need more men and women like them in the future, not fewer.
We hope that some of you watching now will take up this challenge and continue to ask the important questions.
And again, please note that apart from a fleeting glimpse of a petri dish, there is no sign at all, anywhere, of a living cell or a proper view through a microscope. Well, what about this notion of the final frontier? This is what my dear friend David Attenborough had to say from the script provided for him in a recent documentary about caves.
This is our planet's final frontier. And in a world where only the most adventurous dare to go.
Curiously, as I was preparing this talk, on Radio 4 came a man called Bernstein, Professor Bernstein, an American scientist, who actually told the truth of the matter. And this is what he had to say.
Each of us is an ecosystem crawling with microbes, millions of tiny creatures invisible to the naked eye. Aaron Bernstein, doctor and research associate at the Center for Health and the Global Environment at Harvard Medical School, has been studying the ties between the natural world and human health.
Really, we're utterly ignorant about the microbial world. In fact, it is the last great unexplored frontier in life on Earth.
Now that's the truth. That is the truth. And what a wonderful piece of synergy, what a strange coincidence that it should come out just as I was writing those words. Now let's have a look at the BBC's programme Life on Mars. They were looking again at microorganisms in extreme conditions.
NASA scientist Chris McKay has chosen to study life in one of the world's harshest environments, California's Death Valley.
Here in the salt flats of Death Valley, it's not one of the places you'd expect to find life, but since Viking, we found life in a lot of places where we wouldn't have expected. Here underneath the salt crust here, in fact, just a millimeter or so under the surface, a layer of blue-green algae. These organisms are deep enough into the salt that they can access the moisture, but they're close enough to the surface that they're getting sunlight to photosynthesize. What we found, looking at life on Earth in very harsh environments, dry, cold, hot environments, is that wherever there's water, wherever there's some mechanism trapping water, life can flourish. And that's the key. If some alien civilization called us up and said, what kind of life do you have on your planet? My answer would be, it's water-based life. What kind of life do you have?
If water was the key to life, the search for life in the solar system suddenly became the search for water.
Now, that's not what he said. He said, we have water-based life. What, dear alien, what base have you got for your life? He was suggesting there could be lots of others. The narrator then concludes that the search for life becomes the search for water, which is not what the scientist has said. And again, you see a glimpse there of bacteria, just for a few seconds. Let's go to the BBC series The Planets, where they were looking in detail at some of the questions of what microbes are like.
In 1978, a scientific submarine dived to the bottom of the Pacific Ocean and found something completely unexpected.
vehicles like the Alvin beam submersible to hydrothermal vents on the East Pacific rise, and they found these fantastic environments with tube worms and clams and crabs and all these things that were surviving off the Earth's own geothermal heat. And it took a while for the significance of this to sink in, but what it means is that there are other ways of supporting life besides sunlight at the surface of the planet. And that all of a sudden opens up a whole new range of habitability that just wasn't there before.
Now, he's standing in front of a graphic of a photograph of Europa, that great satellite which has water and some people think could contain life. But notice that we saw those smokers, but we never saw what the microbes were like. We saw the man say blue-green algae from the colour in the rock, but you never actually see what the algae are like.
The cave tunnels into the heart of the glacier, where the ice has been frozen for 1,000 years. It's what astrobiologists find in this ice that makes us think that Europa could be teeming with life. NASA scientist Richard Hoover has spent his career looking for life in unlikely places.
Oh, that went very well. Okay, so will any organisms that you find in ice be living in a sense that I would understand it, like they're actually alive now and metabolizing? For a long time, it was thought that ice microorganisms were present only in a state of what is called deep anabiosis, suspended animation. It's now becoming quite clear that that isn't necessarily the case for all the microorganisms. There may be others that are actually actively living in the ice. That's quite incredible. So in this glacier, the whole place, this whole cave, may be populated by living things, not frozen things, but actual things existing, living, cell dividing, reproducing, all the things. Reproducing, all of this.
And there's another case in point. We never see the organisms. It is such a tease. We're led right up to this amazing spectacle, but we never, ever see the organisms concerned. Wherever you look on television, it always happens. I'm going over my time. I do have other examples. Is it all right if I continue?
Oh, yeah.
There is a queue of shuttle buses, engines revving, for anybody who really wants to know. I apologise for going over, but it's such a fascinating subject, and it really ought to be done with some thoroughness. But I do apologise for keeping you from the bar.
This is the cave at the Villa Luz in Tabasco, Mexico, the cave of the House of Light. And it is the definition of a hostile environment to me, because it's full of hydrogen sulfide gas, hence the gas monitor, which says at the moment 1 part per million hydrogen sulfide, very toxic to me, which is why I've got this gas mask in case it all gets too much. So it's a place where you, at first sight, would not expect a great many life forms to survive and flourish. Well, these things are what I came deep underground to see. These are snotites, and you can see why they're called that. They're really one of the most alien life forms that I can conceive of on the Earth, because they metabolize hydrogen sulfide. So they metabolize this faintly acidic and nasty gas that I'm just breathing in now. You can almost feel it on your tongue, actually, the acidity of it. They metabolize it, they react it with oxygen, and they produce sulfuric acid. So their breathing process, if you like, their version of what I do, I breathe in oxygen, react that with sugars and breathe out CO2 and get energy. These guys breathe in hydrogen sulfide and oxygen and produce sulfuric acid.
Now, snotites, named by Jim Solarovich, I think in 1978 or so, of course, isn't mentioned anywhere in the programme. And I love the fact that he called them snotite. It really is for these mucilaginous hanging colonies of microorganisms. A wonderful term. They exist at a pH of less than 1. Less than pH 1. And I was longing to see the organisms. electron microscope picture of the organisms. Absolutely fascinating. I've seen them in the past. But again, we're led up to it, but you never see the organisms. Until I had a visit from an assistant producer working on Richard Hammond's Invisible Worlds. He rang me up later to say, no, we won't actually be able to use you in the program. We're going to do it rather differently. But what they looked at was the structure of the lotus leaf and how it manages to shed water. We saw the structure of the leaf under a microscope. We saw the back end of a spider and its spinnerets. And we saw this.
An invisible world of hidden forces. The machines in here allow engineering on a barely imaginable scale. For some of their work, they need Britain's most powerful microscope. What I'm going to try and do is etch the title of this series onto an area 100 times narrower than the width of a single hair. Type in a small worlds. There it is. That's astonishing. Scientists are only now beginning to understand a snowflake. It's far too small for us to see normally, but these microscope shots show actual snowflakes that have formed a powerful bond. Our world stops there. The eye of a needle held at arm's length is pretty much the limit of human vision. On each tiny toe, there are a dozen parallel ridges. Microscopic photography reveals that the surface transforms into hundreds of thousands of tiny hairs. At 100,000 times magnification, suddenly you can see that each of these 500,000 hairs has itself split ends at the tip, which create even finer hairs. 10 million could fit on a pinhead. These are dust mites. 1 square metre of rug can contain as many as 100,000 of them. In tests, agar dishes placed 10 metres from a sneeze were infected. And they showed that the average sneeze contains millions of microorganisms. A universe of other creatures that we never see. Like the tardigrade. They're the size of a small grain of sand, and they're everywhere. The fresh leaf salad, drizzled with vinegar dressing, complete with a dash of the free-swimming vinegary. And then we come to the cheese course, dusted with a coating of cheese mites. You know that fine grey dust you can make out of older cheese? That's the bits the mites leave behind. But it's not just our food that hosts a thriving microlife. Plankton. These tiny creatures, virtually invisible to us, are an entire universe of astonishing life forms. But their existence is vital to the existence of life on this planet.
Now notice 2 things. Firstly, not a single living cell anywhere. The closest we got were technically rather incompetent pictures of tardigrades, which of course are multicellular, but not a single living cell anywhere in the picture. And hardly anything microscopic. Think of what it was. The surface of a leaf structure, mites, bugs. We looked at snowflakes. All of these specimens were in Micrographia in 1665. And Hooke also didn't look at truly microscopic stuff so much as he looked at familiar things in the dry form. And in essence, what we had is a repeat of Micrographia. The range of specimens in this modern program is the same as in that book from the middle 1600s. And of real life and living cells, there was absolutely no sign whatsoever. For those of you who are interested, since the vinegar eel, the nematode, also appeared in Micrographia, this is what a nematode looks like through a single lens microscope. And please notice that this is a better picture. It's not a very good picture from the point of view of being taken with an achromatic microscope, I agree. But please note that this is really a better picture than any of those you've seen earlier this evening in the other programs that have all looked at what these little vinegar eels are like. And as a result, television misses out on everything. These are some of the standard slides which are here in the teaching lab next door, which I just happened to film last year with my handheld camera, because the silicious structures are so delicious. Gary and I went up onto the roof and we found a pond with some protozoa, and I just took those under phase. In fact, the soundtrack to this is of the class all chatting and laughing and joshing and teasing in the way that we do. But this just shows how easy and simple it is to grab these things wherever you happen to be. And if you click over to the 40 times objective, then you can see the cilia and a good deal of the internal structure of these fascinating little organisms. They are not just boring non-entities and routine non-televisual subjects. They are very, very exciting. Just watch the way. This is one of Dr. Picnic's pictures. Just watch the way in which this wonderful euglena wormed its way around the habitat where it is, exploring. You can see all its inner structure working away, the chloroplasts, which of course give it its source of energy. Watch this wonderful euglena. Look at its sculptured surface and watch its eye as it turns around. Any child can tell you that that's the front end, that's the eye. But this is in a single cell. Yet people who've never seen them still know exactly, instinctively, what it is that they are looking at and what it is that these things will do. I mean, these are such exquisite sights under the microscope, and yet they are simply banished from television. This is a wonderful sequence of this glorious, pretty organism, which so many of us learned about in school, but of course these days they don't. And here, the only organism I know of named after orgasms, Facus. You can see it's a single eye spot in the anterior part of the cell. And look at the wonderfully sculptured cell contents. And watch this predatory ciliate as it sniffs at a pennate diatom and discards it, but it senses this paramecium, it takes a little sniff, and it wolfs it down. All of those actions of approaching it and working out what it's going to eat, what it's going to do, it's just like watching a kitten hunting a mouse, or a bird hunting for grubs. There's all these sort of, all of the cognizance and the recognition and the response and the weighing up of the options before it acts. And only in one place have I seen due acknowledgement of the fact that protozoa are able to conduct sex lives of their own and are aware of each other. And it is, of course, once again, in another edition of The Simpsons, when dear Lisa watches paramecium and sees how even they sometimes can imitate humans in the complexity of their lives.
You don't have to worry about finding dates or dancing.
Oh, yes, they do.
Lisa, they made you some homemade Pepsi for the dance.
So here we have it, ladies and gentlemen. This is what people should be looking at. Just a bunch of euglenoid algae under positive phase in a little limpid drop of pond water. If you turn on television and you have a man saying, oh, which side do I look at? A man saying, oh, is this a dollar sign? I thought it was a yen in an economics program. If you have a doctor, a medical program, and somebody saying, oh, the pancreas, I can never tell those away from the heart. It's embarrassing. Then in sport, if somebody said, this baseball bat is what is used for playing football. I mean, It is at that level of stupidity that these commentaries are being given, and it only happens in this area of science. One doesn't blame the presenters. They are, basically speaking, reading out the script that they've been given. But in an era where we value knowledge, and in the era where bioscience is the leading science of all, it is criminal that the public are kept from this. And if there is anything any of you can do to encourage television people to look twice at what they produce, and to try, as Ted was saying today, to try and get youngsters to actually know this stuff is here, to abandon the tyranny of molecular biology, which has achieved such wonderful discoveries, but now dominates our thoughts. Until we can actually lay claim to the nature of life and the glory of the living cell, humanity will be impoverished. Other countries are very keen on showing it. In the West, we've abandoned it, and it is disgraceful that the media are ever allowed to get away with this dreadful crime against knowledge. Thank you all very much indeed.
We're hiding in the corner.
This really is as close to an English pub as I've ever seen in America. And over here, Arthur Coates.
Trying to be invisible here.
I wonder what...
I almost didn't come back down. Did you see what got in the elevator with it?
Everybody heard, and some were even possibly even in this room, by the Icelandic volcano. It's the first time that a multi-million industry has been cancelled because of something that is extremely difficult even to see. And the story, in fact, is one of mismanagement and administrative incompetence that caused inconvenience to vast numbers of people. It's unimaginable to try and explain to you how serious it was. The whole of Western Europe and British airspace was shut down for several days. It was marvellous. You look in the sky, not a single contract. Live near an airport, it's completely silent. People were saying, why can't we have a week like this every year? It was utterly blissful. But then, of course, you had people checking into airports overseas in Europe to come back with nowhere to go and nothing to do. The companies said that your travel insurance does not cover you because it's not meteorological, it's like an act of God, or Darwin. And it isn't the case that we will pay you. Curiously, my own travel insurance company did cover the delays caused by it. But then they relented a bit, and the airline said, if you send in justifiable bills, we'll do our best to meet the costs. But you had people who had died to get back for weddings and new semesters and all that kind of. I mean, it was a most extraordinary business. And none of it now seems need really have happened. Iceland is a remarkable place. Everybody knows of geysers. And this is Geysir. This is the one that gave the world its name. It, in fact, normally doesn't blow off very much these days. There's another one alongside that is much bigger. But whilst we were there on one of our visits to Iceland, which is an extraordinary country, as you'll see, it did blow off 100 feet in the air. And it is quite majestic and extraordinary. I mean, it is like you think of Yellowstone. But sitting as it does on the mid-Atlantic ridge, Iceland is just bubbling with volcanic energy everywhere you look. I mean, you find entire fields, entire landscapes, which are simply bursting with energy. I mean, there's sulfur in the air, you can smell cinders in the air. The whole thing is a quite extraordinary place. And if you look, for example, this is on the Westman Islands, to the west of Iceland. And here's one of the settlements. And you can see quite clearly, you've got two volcanoes here. And this is a lava field, that has run into the town and is now partly covered by these wild lupins. That's the natural colour of the wild lupin. And these people just live with the imminent threat of massive volcanoes. Here, for example, is a civic building, and as you can see, during the last eruption, the volcano came down, crushed the building, and then solidified. They live with it. It's part of their lives. And to them, the recent volcano was no big deal. It happens all the time. That's what it's like in Iceland. Here, just in front of an advancing wave of lava, is a typical Scandinavian church. And you think, well, isn't it wonderful that it survived? Yes, it's also miraculous, because if you go up close to it, you can see that, in fact, radiant heat has charred all of the paint, and indeed on all sides. So clearly the air temperature was up towards the melting point of the paint. It wasn't just pure infrared from the lava itself. It really is a quite remarkable place. And then you had this volcano, and that's a photograph when it began to erupt. Now, the eruption wasn't the problem. It was the wind direction that was the problem. In Britain, the wind always comes from the southwest. Well, not anymore, it doesn't. We get a lot of winds now from the north and indeed even from the east. East winds used to be quite rare in Britain. You might get, I don't know, 10 days of it a year. Now you can have 2 months where the wind comes all the time from the east. And I keep wondering whether it's because the Gulf Stream is less potent than it was, isn't pumping as much thermal energy into the atmosphere, and to sort of defuse the westward drift. Whether that's right or not, I don't know, but it could be a foretaste of that slow diminishing of the Gulf Stream that we've all been thinking might happen, though hopefully not in our lifetimes. This, then, is the volcano. It is pronounced, and you'll hear it properly pronounced in a moment, Eyjafjallajökull, which to the Icelanders is complete nothing. It's a perfectly normal everyday word as you will hear. It is known to have erupted in the year 920. Much of Iceland, they speak Icelandic, and people say it's a dialect of Norwegian. No, it isn't. Icelandic is the language of the Vikings. It is the Old Norse language. It is the language in which their great sagas were written. And amongst the gene pool, there are a lot of Irish genes. Not because there was a potato famine early on, but because the Irish, you know, you hear of Irish monks going out in their corridors. They were brilliant explorers. Irish monks may even have crossed the North Atlantic to Newfoundland. They were extraordinary people. They made large coracles made of a wooden frame with animal skins, and they could travel thousands of miles. And they travelled right up to Iceland and settled it. I mean, the bravery and the skill of these people is beyond imagination. Then it was well documented in the year 1612 that there was a major volcano. And in 1821 it began to erupt, and each time it died down it burst up again larger than before and carried on erupting for 2 solid years. So we know this volcano, there's nothing new about it.
Back and forward.
All right, well, Iceland.
Right, we'll come out of here. Let me see if I can play it for you directly. Thinking on your feet is something, of course, that you always have to do in these troubled times.
On the very right, Brian. Right hand side?
Far right. Oh, bottom right one, sorry. I may say that it's the first time I've worked on a computer with XP. Do any of you here remember Windows XP? It's an operating system that many years ago they used to use. And as you know, here at McCrone's, we have collections of really old microscopes, really old books, and yes, really old operating systems. Let's see if we can play it direct. It's too good to miss, this particular video insert. No, let's try the MP4 file, which is here purely as a backup, just in case of emergencies. I've been here before.
He's XP'd.
know you can do it.
Fallout, millions of people have been stranded, how it's affecting businesses and how people are coping. Stay with CNN for on-the-ground updates around the world, on TV and online. Coverage continues. CNN.
All right, well, Iceland, volcano, ash cloud, simple words, easy to pronounce in English. But what about this one? There it is. Oh, that's the name of the volcano itself. Not quite as easy. Jeannie Most looks at how journalists covering the story are coping with the Icelandic tongue twister.
While it spewed ashes, newscasters spewed a molten lava of syllables.
Eja, Flea, Jaker, Ball.
Aya, Flea, Yerkabal. It's Aya, Fiat, Yerkel.
Eja, Volek.
That's it?
It's a name so unpronounceable, we can't even tell when someone says it right.
And I'm not going to get this name right, but it's E-F, Yar, Cool.
Most of us are so terrified if it's 16 letters. We avoid any mention of the name.
And the volcano.
That volcano whose name nobody can pronounce.
Saturday Night Live caught on to the way the press is suffering from volcanic naming avoidance syndrome.
By huge clouds of ash shot into the air by Iceland's, I hope I'm saying this right, volcano.
Icelandic interpreters.
The pronunciation is Eyjafjallajökull.
Oh, yeah. Now I get it. The glacier is called Eyjafjallajökull. Am I pronouncing that correctly? Nearly. How do I pronounce?
Oh, geez, it looks like we have lost Al.
See, that's what happens when you try and pronounce the name of that volcano. I said something dirty.
Even showing folks the name doesn't necessarily help.
Eyjafjall.
La. Joe. Q. I don't know. All together now.
Oh my God.
Time to go up to Iceland to get schooled by the natives.
Eyja means island, Fjalla means mountain, and Jökull means glacier.
An easy name to say. Eyjafjallajökull.
Yeah, what English words trip them up?
Pneumonia, or how do you spell it? Pneumonia?
Sounds tired, being weary or something. I cannot say it. I've been practicing for 60 years.
And we had 60 years to learn this.
It's pronounced similar to, yo, I forgot the La yogurt.
Like the La yogurt part.
The La yogurt's kind of close.
The U.S. military has figured out a way around pronouncing it.
Operation E15. E for the first letter of the volcano, 15 for the 15 letters following that.
Can't say it. Make like this opera singer and sing it. Ginny Mo, CNN. What? New York.
We've been singing it all week. I'm Hala Garani. You're watching CNN. I'll see you at 1 p.m. Eastern, 7 p.m. European time. Stay with CNN.
It really is quite extraordinary, is it not, what a mishmash we tend to make of other people's languages. and of course, the person is right. You know, words like cough, thought, and through, and so forth in the English language are unimaginably, unnecessarily complex to find. Well, as soon as the volcano erupted and the southerly flow of air became apparent, airlines knew that you cannot fly if there is volcanic ash in the atmosphere. And nobody had really said what any meant. There weren't actual particulate definitions, nanoparticles per cubic meter or something of the sort. And so things just simply shut down. But look at the extraordinary effect in the volcano itself. If you look, do you see those radiating shock waves that come out? There. Just keep watching those. 760 miles an hour, the speed of sound. You know, sometimes you can see in old Vietnam and World War II movies, you see where the bomb falls and the shock waves, the blast and that. Well, look at them. These shock waves roaring out from the hub of the volcano. Then, of course, you see the red-hot lava and rocks that come after it. Now, that's an extraordinary amount of force. There's a lot of siliceous material. Most of the minerals are siliceous. And the force is enough to shatter them down into extremely small, sharp, glassy particles. As a result, they don't settle out very much. And it was believed that they had a highly abrasive quality. And so everybody said, well, we can't fly if there's any of it. But we were told, for example, in England, that our skies were closed for planes. And yet you'd look up and see a deep azure blue sky. It's not as though there was a haze or smoke or anything of the sort. It was really quite remarkable. Those shock waves are so powerful that you could even get them in altostratus clouds. Look at this example here. Look, do you see the shock waves?
Oh, yeah.
You can see them moving through clouds at 35,000 feet. And that's hundreds of miles away from the volcano. Can you imagine the intensity of power? And that's enough to reduce these siliceous minerals down to particulates that were only a micron or even less in size. And as a result, the legislation began to look ridiculous. There was a report in The Guardian that said that the standards they were adopting meant that there should be no aircraft flying in any atmosphere where there were 10 billionths of a billionth of gram of ash per cubic meter. Now, the original instruction had been to avoid all dust, all volcanic dust of any sort. In April, they changed that and said, well, 200 micrograms per cubic meter might be okay. A month later, they doubled it to 400 micrograms. Meanwhile, you had no actual scientific data to suggest whether this should happen or not. If you look at the maps, you can see that much of the threat wasn't there. Here, red is high concentrations and the dark purple is low. And so, in fact, there, the cloud wasn't touching England at all. There was a very slight raise here. Here, there was nothing significant. There was a tiny little bit of very low concentrations just to the northwest tip of Scotland. I mean, it didn't seem to anybody right to ground all the planes. And then scientists came out and said that essentially these figures, 200 or 400 micrograms per cubic meter, were essentially entirely arbitrary. They didn't actually make any real sense. Yet, between 1981 and 1993, 7 airlines carrying a total of 2,000 people have run into volcanic ash clouds and have been in serious difficulties. But they haven't all actually crashed. And this famous one, in 1982, is amazing. You had Roger Greaves and Barry Townley Freeman. The captain, Eric Moody, Sharon, contain yourself. I know it's exciting. But Eric Moody went to the job. And when he came out, before he went to the lavatory, he noticed that there was kind of St. Elmo's fire outside, flickering lights, like ionization outside the windows. And he thought, this is amazing, I must investigate it. So he went up to the job. And while he was there, the engines, one by one, failed. And as he heard the engine shutting down, he rushed back to the bridge. And the co-pilot said, we've lost engine number 3, we've lost engine number 2. And then he made this amazing announcement. Ladies and gentlemen, this is your captain speaking. We have a small problem. All 4 engines have stopped. We're doing our damnedest to get them under control. I trust you are not in too much. Very brilliant. Now, this was over Indonesia, where this vast volcano had erupted. Nobody knew it had. They flew slap bang into the middle of the dust cloud. And what was happening was that the heat of the turbines in the secondary compressor and in the combustion chamber was melting the siliceous particles, forming a kind of a glass. And as a result, the kinetics of the engines were all wrong, and they shut down. But glass has one extremely important other property, not just that it melts and adheres to titanium, but also that it cracks if you heat it up and cool it down too much. And as they flew lower and lower, they had a glide path, I think, of 1 in 15 or something of the sort, and as they flew lower into cooler air, the glass shattered and began to break off. And so they were actually able to restart the engines. Captain Moody said that since they couldn't see out of the windows, the window had been abraded by silica, and he said it was a bit like negotiating one's way out of badger's arm. So this was this flight, it was 747 from the city of Edinburgh, and lost all power. But they kept trying to restart, and as they got down into the cooler layers of the atmosphere, and we now recognize that the glass fragments just crazed and snapped off and the engine started again. Meanwhile, people behind, back in the cabin, had been writing messages to loved ones on the backs of their tickets and recording little messages, knowing they were going to die. I mean, it's the most extraordinary thing. And these people were extremely brave and skillful to get away with it the way that they did. Anyway, the first test flight this year with the Icelandic eruption, a Dornier from the National Environmental Research Council took off from Cranfield in Bedfordshire, not far from where we live. And a British Airways 747 with Willie Walsh on board, who's their boss, also took off. And these planes flew around up towards Iceland and stuff. They found absolutely no deleterious effect of any sort. They stripped down the engines when they came back. There was no abrasion, no problem, no nothing. The Dornier measured a maximum of 100 micrograms on the flight up towards Iceland and back. And so the revised limit from sort of 2000 or so obviously had to be changed. They reckoned that the cost to British Airways alone was $30 million per day. No compensation for that, of course. The authorities have told you to shut down, and these days, if the authorities tell you to do something, no matter how unreasonable, you simply have to do it. IATA, the International Air Traffic Authority, attacked European responses and said they were thoroughly inaccurate. You know, here you had a little body of people, and everywhere you look, you can be told, no, you mustn't bring that in here because of health and safety regulations. You can't walk up that ladder to change the light bulb because you haven't been trained to do it. Wherever we go, there are regulations that prevent us doing stuff. When it's going to cost a major corporation $30 million a day, then it does begin to get serious. And yet, the people involved, Robert Napier, Dave Hutton, and Andrew Haynes, these are the Quango, this is a European term, a quasi-autonomous non-governmental organisation. These are bodies that aren't elected, but they're set up a bit like the FDA. They're kind of nominated bodies to take control. And when these people were interviewed, these are the ones who issued the edict saying the flights must all stop. They all admitted, we don't actually know anything about aeroplanes. And so what I want to put to the meeting today is this, and it's something, many of you are concerned with maintaining safety standards and quality standards and product standards. Well, what I want to suggest is this, there should be no federal or national controlling regulatory body unless there is a comeback. It should be possible for organizations or for bodies of people or companies who lose millions of dollars because of some essentially arbitrary edict laid down by a non-governmental body. It should be possible to say, okay, we can now scientifically prove that you should not have prevented this, and we sue you for the money that we've lost. You know, when you see signs in a bank or in a shop or in a hospital, the sign will say, we will not tolerate violence or abusive language to our staff. I believe that every one of those signs should have another sign on the back pointing towards the staff that says, we, the patients, will not tolerate any inefficiency, incompetence, or unnecessary delays on your part. And when we have these federal bodies that turn around and say, you must stop flying, they should know that when they say it, the companies can say, well, make sure you're doing it for the right reasons, because if you're not doing it for the right reasons, we will sue the trousers off your buttocks. And in my view, the great lesson of this is not just what standards we need, but the fact that arbitrarily appointed bodies do not have the right, where there is no scientific basis, to prevent companies from acting lawfully. Most of that flying ban need never have happened. In future, it never will happen. And meanwhile, companies like BA are saddled with the financial burden of professional incompetence. In my view, that should never, in any field, that should never ever be allowed to happen again. Thank you very much.
Superimpose that with pictures from the Bud Buffet. Where are you located? I can't believe
I'm sorry. No hair. I didn't know. I think you know what your hair. It's a new demo because it doesn't show up. So when you this one will show up. Yeah, we'll show up. Yeah, I wonder how much stuff is on maybe. Yeah. Okay.
Well, you know, it's funny that you mentioned that because I watched, the only reason I had it was TV before. I don't watch cable. They got beautiful sound and I don't really need a TV. I don't need
That's the next thing, the next film or DVD you might want to acquire in the beginning.
This is what we do when the sessions are over. This is what we do.
That's a nice shot right there. Look at that lighting.
Beautiful.
That's a pretty evil looking guitar he's got. Is that like a BC Rich? Yeah, it's custom. It's heavy, right?
The metal. If you look at them as separately as musicians, they're very talented. Guitar is so good.
Oh yes, trouble
Has arrived.
This is Brian, good friend Brian.
How do you do, dear? Nice to meet you. Carter, the black guy we were just talking to, is just back from a Black Music Awards ceremony in California, where he did very well.
Yes, sir. What he said.
Good night, everyone. Thank you again.
told him they need to pay an H on it for that.
You have the during special.
So, one ball or two. So what are you gonna have to buy?
None. I'm just
to do, because it's the 50th birthday of McCrone research, we're going to do the 350th birthday of the Royal Society of London. It's being celebrated this fall. In fact, I'm very honoured I should be lecturing there this fall. And they had their 350th convocation 2 weeks ago in the presence of Her Majesty the Queen and other members of the royal family, which is also a pleasure to attend. And the Royal Society loves to put itself about as being the longest standing scientific academy in the world, which is not the case. Because 40 or 50 years earlier, certainly 40 years earlier, there was a science academy in Italy called the Accademia dei Lincei, which means the Academy of the Links. Seems an odd title for a scientific body, but it really meant those who perceive like a lynx, those with sharp eyes. An academy of acute observers is what it really meant. But that academy closed down and was reopened under different guises and had a rather checkered political career. It sort of came and went a bit and waxed and waned, as organisations in Italy tend to do. And so the Royal Society's claim to fame is that it is the oldest continuously operating academy of science in the world. And of course it really began with the microscope. We're going to look today at the way in which the microscope, oh by the way, Chairman, what time do you want me to finish?
5, 10 after.
Well, which?
5.
Oh, right. So, it really, in a way...
A few more minutes and be 10 after, really, so...
In a way, it began with Robert Hooke, who was born on the Isle of Wight. Isle of Wight is that little almond-shaped island off the bottom, the southern coast of England, halfway along. It's a tiny little place, and he was born there and came to London, where he became Boyle's assistant. And in a way, Robert Hooke is interesting because he was the world's first ever professional scientist. He was paid a stipend by the newly formed Royal Society, and his duties were to bring forth a microscopical observation at their meetings. And he started in the spring with observations in 1663 of things like moss and other interesting subjects. And of course, he was using a compound microscope for ease. I mean, I don't know whether any of you stop to think why the tube length of a microscope is the way that it is, but it's pure anthropometrics. It is because that is the distance you have to fill between the top of an average table and where your eyes are. Microscope could be any height at all. And so his was constructed by a man called Christopher Cock in London to be a comfortable thing to use. But of course, as I've shown, Hooke also described how you make the kind of single lens microscope that Leeuwenhoek put to such good use, and clearly that's what he used for the fine detail on his observations. But this is the beginning of the Royal Society. They're just starting to have regular meetings, ladies and gentlemen, and here they have a professional scientist whose duty is to bring in demonstrations every time they have a meeting. And it was that sort of involvement of the microscope with the earliest days of the Royal Society, which I find so intriguing. Another fellow of the Royal Society, of course, was Leeuwenhoek. He sent over his observations, or rather he had them sent over for him to London, and they all had to be translated, which took a week or two, and it took quite a time before people began to realise just how monumental were his observations. And he was elected a Fellow of the Royal Society. He was born in the same year, 1643, as Robert Hooke. Of course, he was born in the Netherlands. And when he got to 1686, as Anthony Leeuwenhoek, having been elected a Fellow of the Royal Society, he took the Van as a sort of slightly more noble version of his name, like von in German, like a double-barreled surname in the English aristocracy. And he actually wrote to the Royal Society and said, does the fact that I've been made a Fellow give me a raised order of precedence over clergy in the church when I go on a Sunday? He was very socially aware of the benefits of being elected a Fellow of the Royal Society. So Hooke and Leeuwenhoek, the two great founders of our modern era of microscopic biology were both there at the beginning of the Royal Society. Then we have Henry Power, who was a most interesting man. He also lived in the 1600s. And he was a pioneer of corpusculism, which suggested that everything was made of corpuscles. I mean, in a way, of course, he was actually seeing everything made of cells. But because of the aberration of their early compound microscopes, you tend to see little sort of halos, as you know, diffraction fringes around any sort of particular structure. And so the view began to spread erroneously, not just that living things were made of cells, but that everything, animal, vegetable, or mineral, was made of little corpuscles. But he also wrote some very interesting comments, which sound to me like the observation of Brownian motion. He actually wrote of the solary atoms of light and the constant and tumulatory motion of the atoms of all fluid bodies, which does sound as though he was the first person to observe Brownian motion. Then we had Henry Baker, another fellow of the Royal Society in the 1700s. He was the son-in-law of Daniel Defoe, the great author. And indeed, he edited publications with Defoe. He wrote a terrible narrative poem called A Poem to Restrain the Pride of Modern Man. And it was all about the way that when you look down inside, you see seeds of life and seeds of potency. And in fact, he gave rise to Jonathan Swift's satirical poem about fleas having lesser fleas on their backs to bite them, and so ad infinitum. That was a parody of Baker's poem. Baker wrote a book which was published in 1734 called The Microscope Made Easy, in which he's often been taken to task for having pictures by people like Leeuwenhoek. But if you read the book, it's quite clear that he was acknowledging his sources. He said all the time that, you know, that great inventor, that great discoverer, Leeuwenhoek in the Netherlands, has shown this and this and this. And he was quite meticulous in apportioning responsibility. Then in the early Georgian era, we had Robert Brown. Robert Brown came from Scotland and trained as a medic. And he travelled as a medical man with the 27-year-old Matthew Flinders, when Captain Matthew Flinders went to Australia on a voyage of discovery, and they all brought back, of course, thousands of specimens of previously unseen plants. Brown is known as a botanist, though he was actually a surgeon, but his microscopy was extremely acute. He observed what we now call Brownian motion, the ceaseless jiggling of particles inside pollen grains and also elsewhere in cells. And of course, a sort of casual, an informal microscopist seeing these would think, look at that, I'm looking at living things, you can see the jiggling about, that's obviously the origins of life. And anybody could be forgiven it, but Brown was an extremely Scottish, logical, and objective, self-critical observer. So he also looked in other fluids where you have tiny particles, and eventually he showed that in fluid vesicles in chert, in flinty rock, where you sometimes have liquids entrained, there were particles there which were also jiggling about. He said, well, that obviously is not living. And therefore, Brown in the 1820s, was aware that Brownian motion was a concomitant of the physical existence of nature and was nothing to do with life. We move on then to the middle 1800s, when Sir Everard Home, fellow of the Royal Society, got involved. He was fascinated by microscopy, and he used the Bauer brothers to do drawings for him. Now, this is before achromatic microscopes, and so Home wanted to have the best microscopes available to get the best drawings to illustrate his papers. So he went to the Royal Society to borrow the Leeuwenhoek microscopes that Leeuwenhoek had bequeathed to London when he died. And he took them home and used them to make his discoveries. Remarkable, in the 1840s, the best microscopes available were those handmade by Leeuwenhoek, 150 years earlier. Quite a remarkable testimony. Well, Hone was a terrible plagiarist. He used to publish other people's work as his own. He had the Hunterian papers, and he regularly took the Hunterian notes and had them published as though they were his own research work. And people got more and more cross, and eventually he was told to bring the papers back and return the microscopes. And there was a great fire at the Chelsea Hospital in the apartment where he lived. And Derek de Soller Price said to me that his belief was that the fire had destroyed the microscopes because the microscopes were made of silver. And that may be why they never were found after they were taken by him. The next most notable person to do with microscopes would of course be Charles Darwin, a man who has been exaggeratedly feted in recent years. Now, Darwin was a great microscopist. He was still recommending the use of a single, simple microscope as late as 1858. And he took a microscope made by the same maker as Robert Brown, Banks and Son of London. And he took one of those microscopes because, as you know, they pack down into a sort of little cigar box when they're not in use. And he took one of those with him on his travels. Darwin, it's worth remembering that Darwin was actually an amateur naturalist. He was not trained in the science. He was sent to Edinburgh, in fact, to do medicine, but was such a lousy student that he got thrown out. Came back to Cambridge, where he did a simple degree in divinity, probably based on the idea that you could always look up the answers in the Bible. And he went as a traveling companion on the journeys of the Galapagos, not as their official biologist. They had an official biologist on board. He was there purely for a holiday. These facts, of course, have got muddied and muddled with time. Then we had Lionel Beale, who actually in 1852 started up a course in London on the use of the microscope for students. And William Sorby, Henry Sorby. Now, Sorby was the first person to use the microscope seriously for geological specimens. Robert Hooke had looked at geological specimens under the microscope, but Sorby is the person who actually started doing it seriously. Then William Carpenter, who was a prominent fellow and wrote many wonderful books on the microscope. William Dallinger, who followed in his footsteps, and their books remain collector's items to this day. And then Henry Wellcome, who was an amateur microscopist, made a lot of money out of drugs and founded the Wellcome Institute. In the 1930s, we had Clifford Dobell, who in fact also has resonances of Leeuwenhoek, because Dobell wrote the great biography on Leeuwenhoek, but also he noted a number of amitotic figures in dividing protozoa, which only recently we have realized were true. Lots of people thought they were just artifacts and he got things wrong. In the 1940s, we have Alexander Fleming's great discovery of penicillin being released for the first time. Fleming discovered penicillin in the 1920s, and it was first used by a forgotten doctor up at the north of England who cultured the penicillium fungus and dropped it into the infected eyes of little newborn babies. Then we come to Lord Adrian, who was the first one of the Royal Society's presidents that I ever met. And I've known them all personally since Sir Andrew Huxley, who was the president in 1981 when I discovered the Leeuwenhoek specimen. Huxley did so much work on the diffraction of images formed by the microscope. He was a pioneer of high-power microscopy and did very much work on striated muscle. Then Ellis Coslet, who was a pioneer of the electron microscope. I last saw Coslet, must be 20 years ago, and he was within months of dying. He was incoherent, shambling around his house. But you could tell that he could still understand everything, so I brought him up today with all the gossip. The former president, Sir Aaron Klug, whom I know very well, was a great pioneer of work on diseases like prions and slow viruses. And Max Perutz, who died a year or two ago, I last had lunch with Max about 6 months before he died. He's the man who invented the term molecular biology. He was not only the world's first molecular biologist, but invented the term that has come down to us to this day. In recent years, we've had people like Delpy and Pethica and Williams and G.B. Warren, eminent microscopists who are current fellows of the Royal Society. Athene Donald, who runs a big microscope lab at the Cavendish Laboratory at the University of Cambridge, and Sir Sam Edwards, who pioneered the introduction of environmental scanning electron microscopy at Cambridge, and which brings us up to the present day. And it's so interesting to look back at the history of the Royal and to realise that up to the present day, it is doing great work in microscopy, and yet the microscope was right there at the beginning, the first scientific instrument that anybody had at the Royal Society, and the first branch of applied science the Royal ever embraced. They began with it, and I'm very happy to say, ladies and gentlemen, they're still doing it to this day. There was a new single-lensed microscope with a great wide field of view, just pioneered at Cambridge. They emailed me to ask advice when they were doing it. And I saw it 2 weeks ago at the Royal Society. Princess Anne, the Princess Royal, the Queen's daughter, came and had a look at it with me while we were there. And I said to them, well, it's a remarkable piece of equipment. And she said, I think that's a pretty stunning bit of kit. A wonderfully Prince Annish expression. So the Royal began with microscopes. They're still current with microscopes. And my guess is that the microscope will remain a theme of the Royal Society for centuries and centuries ahead. Thank you very much indeed.
I have a short question with probably a long answer.
I prefer the other way round. You know, where the answer is no idea, of course.
Is there a relationship between the Royal Society and the Royal Microscopical Society? Thank you.
No. No, I should explain that organisations in England become known as royal if the Queen's household considers them to be of great repute. So the Royal Society embraces all the sciences. The Royal Microscopical Society had to apply for the royal accolade in order to get it. And I find it interesting, and you will too, that in England we have the NSPCC, the National Society for the Protection of Cruelty to Children, and the RSPCA, the Royal Society for the Prevention of Cruelty to Animals. Clearly animals have qualified, though I'm afraid mere children as yet haven't. Oh yes, yes, that's right. All of these people, ever since the RMS was founded, all of these people were fellows of the Royal Microscopical Society, as well as being FRS. And there was another point, wasn't there? Yes?
Is the Microscopical Society still a royal society? I know that one of my societies lost it a few years ago when the Crown decided that they couldn't afford all this money in all these societies.
Well, it's not so much affording. The Crown, believe me, can afford anything. But the Royal Microscopical Society, when I joined in the 60s, in fact, almost 40 years ago, you actually had to submit your work and you had to be recommended by a group of fellows and considered by council. And so people could put FRMS after their name, like you would FRS now for the Royal Society. But about 20 years later, they had broadened their criteria so much to embrace almost anybody who wanted to come in. There was no quality threshold. And the letter came from the Royal Household to say, you can no longer use FRMS. You can call yourselves the Royal Microscopical Society. You can describe yourself in writing as a fellow of the Royal Microscopical, but you can't use FRMS because those are academic letters, and your way of recruiting any old enthusiast now is such that it's not special enough to have Royal as an abbreviation. So that did happen, but I hope that clarifies the matter. And you're right, the answer was longer than the question. Thanks very much.
The way that students are trained here, when they come here, there's this great social fabric. And students at MCRI have always learned the sort of communal relationships. In science, people are often very crisp and academic and terse, but here they're warm, they're friendly, but behind it is a steely objectivity. And Walter always told everybody, you must tell the truth the way you see it. Indeed, one of the first graduates went out with a new girlfriend the following weekend, and she tried on some clothes, and she came out with a blue dress, and she turned to him and she said, does this dress make my bum look big? And he said, well, he'd been to McCrone's. He said, why, my dear, of course that doesn't make your bum look big. It's the size of your arse that makes your bum look big. we have the winners. I must say that for 20 years or so, the word auction at this event has always been followed by the word Brian. And this year it was followed by the word silent, which actually means that I don't have to be continually cajoling you to fork out dollars until midnight on a Wednesday. It is such a nice break, I can honestly tell you that. However, the multi-tester was given by Bob Cooksack and has been purchased by Ethan Groves for $1. Ethan Groves is the young man over there with the cropped hair and the bulging wallet. And then that great groundbreaking book, The Particle Analyst, donated by MCRI from its vaults, purchased by Tony Havix for $70. You see, those of you who bought one at the outset would find that its value now will be increasing faster and faster than American investment. But then, almost everything is. Oh, there she is. Kelly, I'm so sorry I missed your lovely talk. But I just took the morning off and went around Old Haunts. And of course, I always hope I can just sneak away, just being just one of the many delegates and just disappear. And then, of course, in minutes, my ears start buzzing. And when I come back, you go, where were you? We were looking for you all morning. Well, I was taking the morning off and having a hell of a good time. Now, Jim McCormick came here on Monday evening. The tousled, white-haired old chap. He was a tousled, white-haired old chap 60 years ago. He just never changed. He has commissioned the manufacture of a number of replica old microscopes and also reprints of early books. And he generously gave a couple of those. They're great collector's items. The one is the Atlas of Microscopes, which has been purchased by Bob Sasha of Cargill for $35. Very good. And remember what I said, everyone's to come. Notes on Nursing by Florence Nightingale. Ed bought that. Where's Ed? Ed Librick. Ed hasn't physically changed, to my certain knowledge, for a quarter of a century.
Should I count Ed? Go ahead. Ed dated her. No, that's not true.
I may say it's an excellent book. The book is written by Florence Nightingale, and it's called Notes on Nursing, and the subtitle is What It Is, What It Is Not. And it was a revolutionary book, an absolute revolution in nursing. And the reproduction is exactly perfect, apart from the fact that on the verso of the title page it says, done by Jim McCormick. But it's a great collector's item, and that's a wonderful thing to have. It's just amazing to see Ed turning up here and still looking the same, because he really hasn't changed, or if he has, it has only been for the worse. Then we've got a PO5 Olympus microscope and an illuminator, been purchased by Susan. Susan has often bought very generously in the past. She has not been inhibited by the need to remain financially solvent. And this year is no exception. $800. Thank you, Susan, very much. A master padlock shot with 22 Magnum, donated by Pete and purchased by Carl. Where's Carl? I don't see it. Oh, there's his hat. The whole room lights up when Carl stands near a light, doesn't it? Carl's bought that for $20. A framed cigarette cards, donated by Bob, purchased by Tanya. Where's Tanya? Tanya goes skipping around this meeting all week, looking like a teenager. You want to tap her on the shoulder and say, are you looking for your daddy, dear? And she turns around and it's Tanya. Tanya hosted me down in San Diego a year or two ago. I must say, the meeting was so beautifully organized. It was so well organized, it was one of the very best I've ever been to. Matched only, of course, by the organization of this one. Number 31, Something, donated by Wynne Hopkins. Lots 31. Well, Wynne gave something to the Institute, which Arthur Coates reckons was worth $5. Oh, wait a second. Yes, it was a $5 bill. Let's Explore Chemistry, donated by Wynne Hopkins. You see, he's given it away. He's so hopeless at chemistry, doesn't even need the book. Purchased by Ed for $15. 2 to go. Heat gun, donated by Win Hopkins, purchased by Martin Kokanda for $2. Oh, make him give more. And then Peter's given a piece of fabric with gunshot residue. He was out in the garage one weekend playing around and foolishly shot himself. And that's been purchased by Ming. Where's Ming Zhu? For $1. That is the lowest bid. You know, I mean, he's an intern. What more would you expect?
Either that or he's Scottish.
May I, on behalf of SMS, I thank everybody. This has been such an easy ride. I so hope this becomes a permanent fixture. I did get roped in to do the announcements, but the value of those goods sold, all donated by people who support us and the way in which students are welcomed here, has raised this year about £2,000. So thank you all very much for your generosity, and see you next year. And with that, the conference closes.
Is there a bus going to leave here to the restaurant?
Yeah, there's a bus that leaves here to go to the
restaurant.
It'll drop you off at the hotel, actually. That's only 2 blocks away.
Should have said something. How many are taking the bus over there?
I'm not sure. You'll have to talk to Carl or to Bob.
The annual banquet tonight is at the Berghoff restaurant, just along the road on Adam Street. It's a restaurant I know well, and it's very, very good.
I would say yes.
A very loud silent action.
And then every now and then, so that's important both so I can for like 4 months because my stuff's not going.
Oh, there's one. Look how cute he is. He's smiling. Such a little pumpkin.
Oh, that's a beautiful picture.
Isn't he a doll?
Yes.
Now see the.
That's what I was trying to say. The 4 is the best one. It's the closest to Adams.
I went in there. Wait.
I got one of those Japanese
And to you guys. People make money doing videos on TV.
Additional. 70's not bad. 100's better. Maybe over 100.
There we are, Dick. You got us both. Great night.
Yep, was.
It was strange that he and I were both saying essentially the same thing, that nothing that we need is being taught anymore. And that is bothersome, isn't it?
Yes, it is.
people have, like the car accident, like, hey, I hit a deer. Well, absolutely.
Yeah, exactly. They don't need to know if
No matter of seconds.
or yeah.
Right.
Okay. So that no, actually it's not. So it goes up a lot of things like that. One guy is up in Canada somewhere. Tire traffic.
No, this was the other one I was looking at.
No one would ever drive in this type of pattern and kind of flood. The only way you're going to get further is in. A lot of people want to put money.
If that happens, most of the time I stop it, I pick up, I move over a little bit, finish it off. I finish off, I leave a spot, but then at the end I go and flip.
I think he's absolutely fascinating. So I'm taking a little speck of hair, because by putting it under the microscope and examining it, I will be able to identify the animal that this came from. Only the microscope can show us these things, and once I've analysed it, I'll know exactly which animal this was.
Projected that well, but feel free to come up and take a look at it. And it's just completely not what you see in a sable, which is a musculid like a wolverine or something like that. But anyway, that's how you clear the doulas. And any questions before you actually start? Here's a section that Brian made of raccoon, and you can see hopefully, yeah, very little, potentially no pigmentation. And as you move through, we gradually are becoming pigmented. You know, in a higher magnified view, and actually seeing that gradual, although it looks pretty sharp when you.
All that cement.
Yes.
Everyone's running the red light. Oh, look at that. Funny. It's supposed to rain. I don't see any sign of it.
Lovely day, isn't it?
I think
they keep saying it's going to go and rain.
I know.
They don't know what they're at, do they, really? It's Peter.
I've got to get one of these. I've seen them.
Oh, the gorilla pod.
Yeah.
That's very good. How are you?
Good. I'm great. And you?
Doesn't he looking good for?
Yeah.
I'm going to tap his hips. There, you hear the metal?
Yeah.
He sets off the...
Hey, Jenny.
He sets off the detectors everywhere he goes.
I do. Can't wait to show him the scar. You said, I think... Oh, you didn't ride it, did you? No.
This is a garage sale or tag sale. And yes, I bought something for Jaddy, but I won't say what it is until we get home, because it's a Prezi. And it was $2. A little later that day, and we're in the hood.
Get your bun charred too.
Okay.
And one with grilled onions and jalapenos and everything else. You have grilled onions or no?
Yeah.
Yeah, jalapenos and everything.
So everything.
Same choices are?
There's ranch, Italian, blue cheese.
Huey's on Balmoral in Andersonville.
And why Huey's?
Huey's because it's one of the top 3 hot dog stands in Chicago. 5-star rating. It's got hot buns. Here we are.
That comment is entirely out of place. True, but entirely out of place.
It's absolutely stupid. And I usually tend not to hear.
This is the silliest thing, Brian. You see the boat? You see the boat down there?
Yeah.
That's as far out as they let you swim.
But see, if you go in here, if you go on this side, then it takes them a while to catch up with you. You know, you can pretend. I just felt like she had a hard life.
Well, she did, and she always played second fiddle to...
Yeah, and I'm sure he let her know it. Well, I don't know how he was personally.
We're now enacting an age-old ritual carried out by the natives of these parts, because Peter and I share an STD. We have a scrotum tightening dip whenever we get together after Into Micro, and today is scrotum tightening dip day in the lake, which is always colder than it looks, isn't it, Peter?
It is. It is.
Yes. There we are. Shoulders down.
Take two.
Shoulders down. He's panting out of this, ladies and
so
gentlemen.
freaking deep out here. It's so deep. My feet are hot.
Beautiful afternoon at the beach.
Yes, very. Now, catch this. How un-European.
I'm going to start.
This is the local police officer checking that nobody has any beer on this beach. I think he's also checking that nobody's enjoying themselves too much, which would also be a federal offence.
We are on the corner of Peterson and Campbell in Chicago.
Yes.
And a friend, woman, was at the butcher shop.
Yeah.
And they've been there for quite some time. She goes there all the time. And there are pictures there of this whole area during the Second War of being garden areas. They call them victory gardens during the Second War.
Yeah.
It came up with the idea of this open space being a garden, approach the alderman.
In England, we call this allotments.
Oh,
because
really?
they allot land to a garden.
Ah. So anyway, that's what all these plots are. But it's taken on a life of its own. There'll be a mural over here eventually.
On the big wall.
Yeah, these are $45 to and they got water in here.
$45 a year.
Yeah.
That's very good, isn't it?
And then they're going to approach the city to buy the lot and then sell it for $1.
Now there's a very, very interesting way of growing potatoes.
Yes.
That's a wonderfully clean way of growing them, isn't it? I do like that.
It is. Can you imagine how easy it's going to be to harvest?
Yes.
The reason for having a plot here is just to socialize.
Meet the locals.
Well, yeah.
Build the community spirit.
It's amazing. When they dedicated this, it was amazing how many people were here.
Oh, now these, and they're quite big too. They're gorgeous, aren't they? Marvellous place. Great sense of community.
So we just put this in. I'm retired. I don't even know the dates.
6 weeks ago, maybe 8 weeks.
Not 8 weeks. Not that far back because the weather was so bad.
Because I know we were out of the country, so we just planted. I mean, we don't need it because we have a garden over in Maplewood, but we did it just to meet people. So your squash is much farther along than ours.
It's had great weather, great growing weather, hasn't it?
Yes, really. You're here from England.
Yep.
There you go. If you don't put this in a place where there's wind and some sunlight and whatnot, then there's some difficulty with the same thing.
So that plant is sweet potato.
This is an eggplant.
Now wait. The sweet potato, that's entirely new to me in a non-tropical garden. That is really quite remarkable. When you put a fork under those, how many sweet potato tubers will you pull out? Just the one?
I believe.
Yeah.
We had a neighbor who lived next to us.
Very nice.
Down the street here for a number of years.
Look at the amount that they've packed in.
Yeah.
You know, some kale and a couple of pepper plants, some tomatoes, and at the back, a line of about 8 sweet corn.
Jeez.
What about
look at the
homegrown watermelons?
Look at the yield on this tomato so far.
Yes, that's a good crop of the plum variety from one plant. Pretty good crop of sweet capsicums there too.
Look, and this one.
An eggplant next door.
And this one already has eggplants.
Look at that. Good stuff.
Blueberries. I mean, who would have thunk someone would put blueberry bushes in a garden?
Well, well, well.
Blueberries.
And the noise of the cicadas. There's all this cicada noise, and cicadas, as far as one knows, don't migrate. Why don't we have cicadas in Britain? I mean, our winters are not as cold as yours, and our summers are not as hot.
Well,
but basically speaking, the climates are equivalent in many ways.
Oh, it's much hotter here.
Oh, it's hotter for longer too, isn't it?
Yeah, and hotter for longer, and colder for longer.
Ah, yeah, that may be it. And Peter's now mature garden. The garden is a delight.
Ah, good. It is.
The bathroom needs a little sprucing up. With the exception of, this is my lovely kitchen. Hello, Jen.
The sun deck at dusk.
No.
Bread. That trumpet vine survives out here in the rigors of the Chicago winter, so it might be worth trying at home.
Let's eat. Please.
You'll have to see the movie. That's the only reason I got these.
Okay. We'll do that.
This is lentil salad. It's a few days old.
Thank you very much. Can I offer you some buet, Pierre?
I ate that a long time ago.
It's all homegrown.
Yeah.
That looks marvelous.
This is a French lentil salad. It's not, I didn't make it today, but it's still good, the salad. And there are beets. This is ricotta mixed with, for your bread, I think it'd be nice. Ricotta mixed with sun-dried tomatoes. And then this is a manchego, this big chunk of cheese. Okay, with truffle, I think.
Okay.
And the other one is brie with apricot and nuts. I'm gonna go red. You.
Oh yes, of course. You're very, very, very good health.
Likewise.
To many more evenings thus.
Yes.
Absent friends.
Jan, wish you were here.
We sure do.
I wish you were here too.
Ah. Let's not show them me. Really nice. God, even with ricotta, that sun-dried tomato is strong.
What's sun-dried?
That would be disastrous.
But it would be fun though, wouldn't it?
No, not for Peter.
Eventually, I might have to ask her to leave.
Yeah. Eventually. Monday, noon. Out of here.
How close can you get with that?
This is Irving Park. This is a Blue Line train to O'Hare.
Przejście do wyjścia M2, nasz samolot gotowy jest do odlotu.
bread, we have some freshly made bread, parmesan and sun-dried tomato, and we have a really Caesar salad, homemade Caesar salad.
The house and the Jaddy look wonderful.
Hello.
Hello, sweet.
Love you.
Well, and after 30 hours of travelling, to come home to this beautiful house, this beautiful wife, this gorgeous meal, and presents.
Yay.
Which, oh, yes.
For Mum and Dad.
Oh, excellent. Short pyjama set.
Yes, but what's the most important thing? Not that they're short.
Oh, no, but it's The Simpsons, of course.
Yay.
But I like short pyjamas. I have no idea what this is in here. This is an actual properly wrapped something interesting, kind of thing you'd hang on the wall.
Isn't it, Jeff?
Let's have a little look here. Oh, I say. Oh, a seaside arrow to go down by the summer house, which is going to be our seaside chalet. I was going to have to make one of those, and it would have been nothing like as good as this. How gorgeous.
Yay.
And we'll make sure that it
home.
points exactly to the beach. I love the fact it's got this statutory label saying it shouldn't be played with by children because of small parts. Because the sign was a couple of feet long, and the only small part is the label.